Okay. Thank you, Kai-Fu, and thank you, Liang, for that wonderful introduction and for giving me this opportunity to share with our work, share with you our work. I think this is my first time presenting to this audience, so it's definitely excited, and it's nice to meet you on Zoom. Um, so I'm presenting to you this work that was many years in, in the making. So this is our attempt to analyze single cell and spatial nanopore long-read sequencing data, trying to identify spatially and cell type specific, spatial specific and cell type specific alternative splicing. So I'm going to try my best to convey this work to you. I just want to start off because I'm not sure how much time we'll have at the end. I want to start off by acknowledging the main drivers of this work. This work was started with a collaboration with Han Ji, who's my collaborator from Stanford. He started generating this single cell long read data, and that was very exciting. And then Yun Tian was just joining my lab as a rotation student when this lab started. And he came with a lot of good ideas and his own interpretation of this data, and that morphed into this project. So a lot of what I'm going to tell you is actually ideas generated by Yun Tian. He's the main architect. So he's here to help me if I get into trouble and forget the details. And then at that time, the postdoc at Stanford in Hanli's lab, who did most of the wet lab experiments, is Hong Sung Kim, who's now a faculty member in Korea. So these are the three main drivers of this work, and there's a lot of other people involved, but I just want to start by this acknowledgement. So, you know what alternative splicing is. A gene can have multiple isoforms, right? And even within individual cells, there might be multiple isoforms of the same gene. Now, when we do library preparation, we extract the RNA and then we attach UMI, which is the unique molecular identifier, which is now common to all single cell and spatial barcoded experiments. And then, the UMI barcode and maybe the single cell or spatial barcode, that's the library preparation step. And then the standard short read sequencing data would just, give that to the Illumina sequencer, and then you will get the tag counts. And that loses, of course, this isoform information, right? So the standard short read pipelines only give you the ends, and then you want to know what isoform of the gene is being expressed. And that actually matters a lot. We see, this is from our data, we see that there can be genes whose total expression right here does not change between cell types. Say there's two cell types in this example. However, the isoform level expression does distinguish between cell types. So here you have isoform and one isoform two. And wouldn't it be nice if we could actually profile the isoliform level expression in a high fidelity manner? So even more, this is, I think, a common knowledge in this group, that differential, splicing really has the high importance when we want to distinguish certain disease cells. For example, here's the cancer. A differential splicing event that happens in cancer. This PKM gene, which is part of the oxidative phosphorylation pathway. It is a pyruvate kinase. This has the PKM1 isoform in normal cells. However, in embryonic stem cells and also cancer cells, it switches to PKM2, and that slows down the pyruvate kinase step and diverts the energy metabolism to aerobic glycolysis. So that's really functionally relevant. So with that, so what Han Li's lab tried to do was, instead of plugging this single cell or spatially barcoded library, cDNA library, instead of putting it into Illumina, for nanopore sequencing, there were some detailed library prep that actually matters here that's different from Illumina, but I'm not the experimentalist here, so I'm gonna just show you this simplified picture. So Nanopore is a long read sequencing technology, allows you to see most, if not all, of the isoform. However, we started this this project back in 2000, like 2000 and 2001-ish. And that at that time, the nanopore sequencing was very error prone, right? So that was an R9 chemistry. So a lot of our early efforts actually was in dealing with the fact that there's so many sequencing errors in this that it causes, as you'll see later, misidentified UMIs and cell barcodes and things like that. Now the chemistry is much better. Now it's about 99% accurate. However, as our benchmarking will show, that there are still the artifacts that we have discovered in the low accuracy phase of nanopore sequencing still carries forward. And I'm talking about this in abstract. I'm going to go into the details in just a bit. So first, a sort of high-level picture. So we have long read sequenced single cell or spatial libraries. And then what we have developed, what I'm going to show you is a pipeline, a computational pipeline for accurate single cell isoform quantification. And with that, that allows us to do many downstream analyses, but we specifically focus on two. One is to quantify isoform heterogeneity at single cell level. Now I'm going to go into detail what that actually means. The second is to do differential testing, so identifying differential splicing events between cell populations. Okay, so with that intro, here is an outline of the rest of the talk. So I'm going to show you how, in terms of single cell isoform quantification, I'm going to show you how does sequencing error affect quantification, and then how we address it. And then I'm going to show you some benchmarks. And then we are going to look specifically at what I think is a very statistical problem of how to quantify isoform heterogeneity at single cell level. And then the second statistical problem, which is how to do differential splicing detection between cell populations. Okay, so okay, first question. How does sequencing error affect quantification? To talk about this, let's look at the design of a read, right? So in a typical read, a typical nanopore read, there's this part at here where it is actually containing the isoform that you want to know about. So the exon 1, exon 2, exon 3, poly A tail. And then during the library prep step, what's attached to it is an adapter, the unique molecular identifier, which is in green here, and the cell barcode, which is in blue, right? So this is the part that is different from single cell and bulk sequencing. So actually, lots of people, what they do is they try to use very simple scripts to extract the UMI and cell barcode. And there's other papers, other groups have worked on this problem. And once you have extracted the UMI and cell barcode, then you can just plug your data into a bulk level tool, right? So I hope one of my goals is convince you that this UMI barcode is actually very useful. It's a source of distress for us for single cell isoform quantification, as well as a source of opportunity. So I'm going to show you exactly why that is. Okay. First, why is it a source of distress? Why is the UMI barcode more difficult to analyze here? It's because the UMI barcode, it's a short barcode sequence. And we basically, in single cell and spatial data, you count the number of copies of the gene by counting the number of copies of unique UMIs, right? So let's say here you have 7 reads, right? But you only see 3 different UMIs. Then there's only 3 copies of this gene. However, because of the noisiness of nanopore sequencing, you don't get the exact UMI. When you do Illumina sequencing, it is so accurate that you can disregard this, right? But in nanopore, you will get once in a while, with 1% chance, you're going to get a UMI that is not actually a true UMI. It's actually a spin-off of this cluster. It's actually the same UMI, but you think it's different because it has these sequencing errors. So this is what we call scattering. UMI scattering is where you think you have more copies of a given gene, not really more copies. It's because your UMI is corrupted. Okay, so how serious is this problem? I mean, at first we thought, okay, maybe it's not that serious because, the errors are rare. So, how rare are errors, by the way? So we can, it depends on the data set. It depends on your actual experiment, right? So Nanopore publishes these error probabilities, but that's under ideal settings. For your experiment, we can actually get a rough estimate because we can extract the Illumina R1 adapter sequence, which we know what that sequence is. We know where it is easy to find. So we can get that sequence and look at how many different versions of that adapter sequence are there in your data and compute the edit distance. And that gives you a sense of what the error probability is. And then we can simulate data to see how much how much UMI scattering there is. So, this is a graphical depiction where every dot is a UMI and they are colored by their original true UMI identity. So of course it's simulated data. So we know exactly what the true UMI is. So then we can color by it. And you can just see, what happens is that, this, for example, this blue gene, UMI, actually gave rise to 3 clusters. So its count is inflated by 3. So I'll show you exactly how much this matters in a bit, but I just want to digress a bit and look at a different problem, which is just as serious. Here, I'm talking about misalignment. So this is an example of a gene, RPL41, that has a short exon in the middle, and that is differentially spliced. However, you see that these 2 reads in the middle, where you think that they don't have this exon, they're actually mistakes because you see that they have this region here that has a lot of errors, a lot of disagreements. Well, that actually should have been here. They are wrongly mapped to that end because of the fact that there might be a few sequencing errors in that sequence, and that misguided the mapping. So they're wrongly mapped segments of the read. So you think that is a differentially spliced isoform. The reason why we know that this is not is because in the annotation this isoform doesn't exist, and also the fact that you see so many errors here in all of the instances where you have this exon, this short blip right here, that makes you think that that's a mapping error instead. So, with more sequencing error, you're going to get more of these kind of events, especially affecting short exons. And here is where I'm going to show you that you actually have an opportunity to correct these in single cell data, whereas you couldn't correct them before in bulk sequencing data. In single cell data, you have the UMI. The UMI, if correctly recovered, can actually help you correct these errors within each UMI cluster. So more on that in a bit. So I just showed you how does sequencing error affect quantification, mainly in 2 ways. One is misalignment due to low quality sequences, and the other is UMI scattering, which is the effect that when there's errors in the UMI, it leads to an over quantification of the gene expression. So with that said, I'm going to show you the entire pipeline of long cell, which is this tool that Yuntian developed to deal with these 2 issues. And it's a long pipeline, so I'm going to describe it to you in parts, okay? So first, let's look at, well, first, I just want to show you, all of this is using as, a toy example, this truth, where there, you can think about what I'm showing you is a gene with two isoforms, A and B. A has a short exon in the middle that can be spliced out. And then let's just say that for this gene, there is also a possible misalignment, which is denoted by N here. Okay, and that is wrong because in this toy example, we know about everything about this gene. So the first the first step you do is obvious. You have to map the reads to the genome. And then after mapping, or concurrently with mapping, you identify the cell barcode and UMI from the read. Now that actually took us a long time to figure out how to do, and other groups have worked on this problem and found good solutions for it. So I'm going to skip that step because I think that is a solved problem now, how to identify the cell barcode and UMI from the long read. Okay, after you do that, then what we do is UMI clustering within all the reads that map to the same gene. Okay, so you basically, this is based on a graph, we use a graph-based clustering procedure, basically to identify those UMIs that are within a small Hamming distance from each other. And so, yeah, okay, so you identify the UMI, and now we have these UMI clusters. Let's say we have 3 clusters for this gene in this specific cell. So this clustering happens within the cell. And now, this is a very important step, is misalignment and truncation correction. Misalignment is a problem I just showed you. Truncation is a common issue in nanopore sequencing because oftentimes there's, early truncation, so you don't actually get to see the entire isoform. So you would like to correct that mistake as well. So what we do is this iterative, heuristic procedure where the rationale underlying this is that for all the reads that belong to the same UMI cluster, they should come from the exact same molecule. So they should agree completely, right? So if you have a situation where, like in cluster 1, where there's 3 different reads that don't agree with each other that have the same UMI, only one of them is correct. And if we think that the error probability is low, then what we expect is that if we do a majority vote within cluster, we should get the correct isoform coming out of it, right? Because we assume that errors, truncation errors or misalignment errors. So this is a truncation error, and that is the N is a misalignment error. These should be rare. So what we do is within each cluster, we identify the majority, the one, the isoform with the highest count. And then we do this attribution table where we assign other isoforms, other putative isoforms, maybe wrong, to the isoform with the highest count within each cluster. So, B is easy because, they all get assigned to B. And then A got assigned to itself in this cluster. And then, it got assigned to itself here as well. So there's 2, 1, 2, 3 that it got assigned to A. And then this N got assigned to A because it lost in that majority vote or plurality vote. Okay, so of course, this intuition of the wrong isoform being lower count than the correct isoform may be violated just due to sampling error, right? Because you say the error probability is low, so that should be the way it is. But sometimes with low counts, you can still have the wrong isoform being the one with the highest count. So what we do is we sum across cells. Then once we sum across cells, law of large numbers kick in, and the correct isoform will win most of the time. So, so this allows us to harness the UMIs that are unique to single cell and spatial experiments that was not in the bulk setting. We can harness these UMIs, these UMI clusters to correct misalignment and truncation errors. And that is something that we think, makes this, even better than the bulk setting. So, after truncation correction, we have two UMI. So we would think that there's two counts of isoform A and one count of isoform B. Okay, so how much does this really help? You, so how how often is there a misalignment error that can screw your results, skew your estimates? So to answer this question, we looked at the bulk level percent splice in that is estimated for constitutive exons. Now we know that for constitutive exons, they should have a psi, this percent splice in of 1. That is the known truth. This is real data. We know that for these constitutive exons, we should get a psi of 1. However, you see that in the raw data, due to mapping error, the psi is actually much lower than 1. So that difference is due to mapping error. So you see that after we corrected by this procedure, we push that up to 1. So it's much better. And here's just one specific example of this VPS28 gene. And this gene, I think this is a constitutive express exon, so it should always be here. But sometimes it gets wrongly mapped. This is exactly the situation I was showing you, where a small exon gets wrongly mapped because of sequencing error. And that is shown in the pile of plot as well. So after you correct it, you don't see that anymore. And if you compare across different tools out there, they don't make use of the UMI clusters to correct this mistake. And they actually, like for example, IsoQuant, which is a commonly used tool, actually get it wrong almost 25% of the time. So that's the, step 3, the misalignment and truncation correction. And then what we try to do is to fix the UMI scattering issue. The UMI scattering, remember, would create false isoform cluster, false UMI clusters. So you would say, for example, this is a singleton cluster. This cluster 2 only has one read with that UMI. And that got filtered out. And I will show you let me just skip ahead and show you the intuition behind this scatter filtering step. Okay, so in terms of UMI scatter filtering, the intuition here is that each isoform should have a similar amplification fold when you apply PCR. So in single cell and spatial experiments, there's a PCR amplification. And the amplification efficiency actually varies across different molecules, right? It depends on what the initial molecule is, the amplification efficiency can vary quite a lot. However, the same isoform should have the same amplification fold, which means that if you look at the number of reads assigned to each UMI for the same isoform, that number, what we call the UMI cluster size, should be approximately the same for reads that represent the same isoform. And we can see this in real data. So this is this is in data where we looked at four genes that have the same that have only one isoform. So because they only have one isoform, we know that we can just look at the UMI cluster size, which is a proxy for the PCR amplification fold, and plot that against expression. So each dot is a cell expressing this gene. And you see that indeed, the PCR amplification fold is about the same across different cells, regardless of their expression level for this gene, which means that we should have a common estimate for the PCR amplification fold across cells, across expression values, expression levels for the same isoform. Okay, so that gives us a way to identify scattering and remove it from the data. So what we do is we take the adapter sequence again, and then look at the Needleman score, which gives us a sense of how frequent the scattering would be. The frequency is here 6%. And then we would, for each gene, we would order the UMI size, cluster size, for each isoform, because now you have mapped it, so you know the isoform, the putative isoform. You would order the UMI clusters and you from smallest to the largest. And the hypothesis is that these small clusters of maybe singletons or maybe two reads, these are the scattering. These are due to sequencing error. So we would successfully remove them until the number removed is greater than the expected number of such clusters, given the total expression value of these remaining clusters. So it's an iterative procedure. So in the end, for this example, your UMI count will be 4 instead of 6. So this really makes a difference. Here, I'm just showing you the PCR amplification fold versus the quantification level for a simulated data set, simulated based on known error probabilities in Nanopore. And you see that this is cluster, this is scattering, right? It should be 1. The simulation is that we only have 1 copy. However, if you don't control for it, the quantification will be skewed up, right? So this is, scattering in data. And then here, so this is simulation, but this is real data. In real data, you see that we had a single cell data set where we sequenced both using short and long read. So 10x is short read, this long cell is long read. And then if you see that if you don't do this scattering correction, which is this DP scan procedure, the baseline procedure, you will see a lot of these small UMI clusters. These are the errors, and they shouldn't be there. In fact, the reason is because we're only comparing for genes that have only 1 isoform. So short and long read should give you the exact same UMI cluster sizes. And after we correct for it, we get matching distributions between short and long read. And the reason why they should be matching, again, I just want to emphasize, is because these genes are not supposed to be differentially spliced. So how does long cell address these 2 different issues of misalignment and UMI scattering? We have these 2 steps in the procedure. One is using the attribution matrix for correcting misalignment, and then we do this UMI cluster size scatter correction. So these 2 steps we feel must be done for single cell and spatial experiments. And because of this unique way that experiments have these UMIs, and you correct for PCR amplification error. And also, I just want to emphasize that in data, we don't know what the PCR amplification fold is for each gene. And we don't, so you see that this noise property actually depends on the gene's initial expression as well as its PCR amplification fold. And those are 2 things we don't know. So it's very hard to correct for them downstream. So you have to do this in the pre-processing step. So I'm going to show you some benchmarks. First, we did a lot of simulations. In simulations, you know exactly what is the expression level at the isoform level. So we simulated, this is a pretty complicated procedure. We simulated data to have similar sequencing errors and truncation error and mapping error of real data sets. And then so it has these steps that try to mimic a real experiment. And then we know the ground truth, of course, because it's simulated. And then we compare whatever method it is to the ground truth. And then you see that in the simulation data set, we tried it for 2 different scales, 1,000 isoforms across 187 genes, as well as a larger data set. And the baseline for these data sets, the simulation parameters for these data sets are estimated based on real data. So these are, that's why these numbers are, are like that. So you can see that the per cell correlation between the estimated and the truth increases when you do those scatter correction and misalignment correction steps in long cell. We also tried pairing long cell with this isoquant, which is a tool that was mainly motivated by bulk sequencing, but they have this way for you to input these single cell level count matrices, the single cell level reads to them. So we can sort of couple that, adapt that to our method, and then we see that in the simulations, it gives an even further improvement. Here is real data, not simulated. We took the cell line. This is a Jurkat cell line. I think it's a melanoma cell line. And then we constructed 10x libraries for this. And then in here, it's a targeted sequencing. We targeted 110 isoforms in 18 genes. And then we plug it into two downstream protocols, PacBio and Nanopore. PacBio is supposed to have, in some certain ways, better properties, more accuracy, and such. So we can use that. So IsoSeq is PacBio's own pipeline. So we can use the output from that as a gauge on the accuracy. It's not perfect because PacBio has a lot of its own errors, but it's the best we can think of. So by this metric, we see that Cisalor 2 and long cell do pretty well. Long cell is slightly better, but in this benchmark, somehow pairing long cell with isoquant couldn't do as well. And then the final benchmark we did was this spatial long read experiment where we, this is a public data. And I forgot to put the citation here. So in this data, they looked at the olfactory bulb, and it's VCM. And then with the VCM cDNA library, it's sequenced by either Illumina and Nanopore. And the special thing that they did for this data set is that they really did saturated sequencing by Illumina, meaning that they were able by Illumina to basically capture every single UMI in this origin cDNA library, or so we hope, right? Because then we can use the Illumina UMI as a whitelist to guide the UMI recovery for Nanopore. Now, note that we cannot do this usually because it's very expensive to sequence Illumina at such depth. But anyways, for this data set, they did it. And then so we can use that as a sort of gold standard. Not at least for the UMI scattering correction step. I mean, still you still don't have misalignment errors are still there in this, but we can use that as a gauge. And then so looking at the correlation between the estimated versus that using the original the Illumina guided UMI procedure, we see that with long cell you do pretty well. This WF single cell result, this is Nanopore's own pipeline. So that came out while we were revising our paper. So they do pretty well too. Long cell isoquant actually does the best. So you see from the benchmarks that we have a pretty robust procedure to do quantification of single cell isoform expression. So up to now, any questions? Okay, so I'm going to now go on to some downstream analyses, okay? The first question we wanted to ask was, how do isoforms distribute among cells? Now, what does that exactly mean? So let's say that you have a gene that has two isoforms, isoform 1, isoform 2, and let's say that in bulk sequencing, you saw that it's about 50-50 between these two isoforms. Then the legit question to ask at the single cell level is, how do these two isoforms coexist within or between cells? There could be one scenario where you can see that both isoforms, isoform 1 and isoform 2, are in every cell. So each cell carries both isoforms. Now you can imagine that has a biological interpretation, that these two isoforms are both performing functions that are needed for each cell. Or you can imagine another scenario where the two isoforms segregate into different cells. So a cell either splice one or express one or the other, right? So individual cells explicitly express one of the two isoforms. Now this is called high intercell heterogeneity. And then on the left here is high intra-cell heterogeneity. Intra-cell meaning there's high heterogeneity within the cell. Here is high heterogeneity between cells. Both have their biological interpretations. So anyway, so we're going to characterize this by using this phi, it should be phi sub c. So phi c, there you have a phi parameter for each cell. And that is the ratio of the molecules within that cell that has this yellow exon spliced in. So phi vary from 0 to 1. So you see that in a case where there's high intracell heterogeneity, phi is at 0.5 across all cells. Whereas with high intercell heterogeneity, phi is at 1 for some cells and 0 for other cells. And then so if you do a histogram of phi, sorry, I've been saying the wrong Greek letter, psi. Okay, so yeah, because percent PSI, percent splice in. So psi, so psi could be a sort of like a unimodal, right? So at this mean level, so that means that there's high intracell heterogeneity, or it could be sort of bimodal, right? It could be shaped like this, like a U, that's high intercell heterogeneity. Now, remember there was this paper a while back called, I think they had this pipeline called Expedition for looking at single cell splicing, where they basically concluded that basically all cells are spliced all genes are spliced like this, that there's high intercell heterogeneity. There's very few genes that are where you have two isoforms coexisting within a cell. We found that to be, spoiler is that we found that to be actually false. That oftentimes you see high intercell heterogeneity in your data. That's not because the psi is truly different. It's because of low counts. So you have a lot of sampling error, sampling noise contributing to the fact that you think that it's like this, where it's actually like this. So, but we would like to control for the fact that you have the sampling noise, right? Because for lowly expressed genes, you're like, looking at a binomial with small total count. So we model the psi c. You don't see psi, right? What you see is the total and the number of splice. So psi c, if you were to see it, you would model it as a beta distribution. Now that beta distribution would have mean and a variance. And the ratio of the variance divided by mean times 1 minus mean is this parameter, which we call psi. Now that psi is the dispersion parameter. Okay, so every beta distributed parameter can be parameterized by alpha beta or by mean and psi. So mean is basically the bulk splicing level, and phi, mean of psi is the bulk level, and phi, which is the variance divided by this expected variance, is the dispersion level. And then so you can actually estimate, I didn't show you how we estimate it. It's very easy. It's just maximum likelihood. You can estimate psi and phi for every single gene isoform across cells. And then you could put that on plot that, right? So here I'm just showing you a cartoon of what it should be. Like if psi bar is 0.5, meaning the bulk splicing level is 0.5, then a low phi means that there's high intracell heterogeneity, means that you should see this kind of curve distribution. If phi goes to 1, you should be seeing this kind of U-shaped, right? And then the bulk psi level can shift also between 0 and 1, and you see different distribution patterns. So I want to emphasize here is that if you fix psi bar, if you fix the bulk expression level, then there is an explicit trade-off between intercell diversity. And you can see that here, right? So you can think about if you fix the sidebar level, meaning you fix the proportion that goes to isoform 1, isoform 2, and you have these end cells, then you can think about how to divvy them up across cells. You would be either increasing intracell heterogeneity or increasing intercell heterogeneity, depending on how you divvy up. So there's explicit trade-off. Okay, so in terms of how to quantify isoform heterogeneity at single cell level, we developed this, statistical procedure, and then we can apply it to data. So let's look at a few data sets. Here is a colorectal metastasis to liver. It's a single cell data. There's macrophages, epithelial, pretty standard data set. And then you can, for each gene and each isoform, you can estimate the sidebar as well as the phi. Remember, both are values between 0 and 1. So this is within the unit square. However, you don't see very high values of phi. So, you see these. And then I want to forgive this non-traditional graph. We are using the color to denote the confidence in that estimate. So we can get a confidence interval. So, so you see that so I see this as akin to a highly variable gene analysis. In single cell and spatial data, you start off by looking for the highly variable genes. These are the genes that are informative for cell type or spatial structure, etc. So the analog of that in single cell and spatial splicing analysis is this phi. If the phi is high, that means there's high intercell splicing heterogeneity. If phi is low, there's high intracell splicing heterogeneity. So here, let's look at this MyL6 gene. You see that this remember has a high phi, so means it has high intercell heterogeneity. Here is what the estimated psi looks like across cells. And here is the UMAP of how the psi is distributed across cells. You see that this is a epithelial specific isoform, right? So basically by computing these values, you can quickly zone into those cell type, probably cell type informative isoforms. And here is just the sashimi plot that shows you how this specific exon right here on this MyL6 gene is the one that is differentially spliced between epithelial and immune. Just as a counterexample is this RBIS gene, which is low phi, I'm sorry. And you see that, yes, the histogram is sort of unimodal, but you do see a lot of cells here that have none of this one or the other isoform. That's due to sampling. Okay, at least we think that's due to sampling because we cannot rule it out from from sampling. So you see that there's just not enough evidence that this is cell type specific. So just another example, I just want to, here is a brain development data set that goes, you see brain cells, neurons going from neuroblast to mature GABAergic and glutamatergic fates. And when we compute the sidebar and phi, we see a distribution, and we want to focus on those that have a low CI, meaning a small confidence interval. You see that PKM, for example, here has a low CI. A lot of them don't have really high confidence because the gene is expressed too low, but we just added them here anyway. And then you see that PKM, which is this top gene here, is a splicing isoform that is informative for the stage of differentiation into the glutamatergic arm. So remember, this is the stem cells, and it goes like that. And then you see that it has this U shape. Okay, so this is a quick way to pull out those isoforms that are intercell differential or intracell differential. Okay, so this is an intracell differential spliced isoform. When it's intracell differential spliced, that means that both isoforms of this gene must be somehow important for the functioning of the cell. Okay, so one thing we realized when we applied this to data is that there's universal high intracell splicing heterogeneity, which is not what was I think what was believed before. So you see that there's a lot of highly confident intracell spliced genes. These blue dots here, that's where with very low phi value. Okay. So I think I have time maybe just for one more example. Here is, I just want to show you a analysis where we try to identify differential splicing events between cell populations. Let's say you have two populations, population one, population two of cells. Okay, and you want to find differential splicing. So what you observe is the count of reads with a given exon, which we call EI, and the count of the reads for that gene, which is GI. So you want to know, what is the percent spliced in of that exon, which is this phi. Okay, so your observed data is actually these scatter plots of gene count versus the exon count. And this underlying C is what you want to compare. Now, our procedure actually allows you to, by maximum likelihood, estimate this percent splice in. But in terms of comparing the distributions, remember we have a distributions now because you have a psi C for every cell. For every cell, you have a estimate of the percent splice in for that cell. And across cells, it becomes a distribution. And this becomes a test of the difference between two distributions of psi C. So when comparing those two distributions, you can compare them in terms of their mean as well as their dispersion, right? So those are two ways you can compare distribution. So we developed likelihood-based test for that, which is easy. When you think about it, it's just testing between two beta binomials. And then we can, pull out genes that are different, that are sort of, differentially spliced in terms of the mean or the variance. And this procedure accounts for uneven total gene expression across cells. So it could be that cell population 2 expresses a much lower count for this gene, right? And that shouldn't affect your estimate of psi, or it shouldn't affect your hypothesis testing of psi. So this procedure accounts for that, and it sensitively detects both a shift in mean as well as a change in dispersion. So finally, we applied this to a CRISPR knockout experiment conducted by Zhang. And he basically used a CRISPR to knock out these genes, which are splicing factors. And then in this Jurkat cell line, this melanoma cell line, and then he also stimulated the cell line and also did the same experiment, just for the heck of it. And then so within this experiment, you have the counts of cells that got the gDNA for a given CRISPR knockout. So you know how many genes got these splicing factors knocked out. And then you could do a differential expression. So it's an exciting experiment. However, I have to say this experiment is very low powered because, CRISPR perturb-seq experiments are notoriously, low signal to noise ratio. And we have too few cells here to really make a lot of discoveries. However, we did make some. So we applied this procedure. We identified, because another thing is because this is a full transcriptome sequencing. So we're testing across all the genes. So you have to control for multiple testing, right? So after all that multiple testing, FTR correction, we got this many genes that were differentially spliced with the CRISPR knockout of one of those factors in the Jurkat experiment, in the original cell line, as well as the stimulated cell line. And what increased our confidence in terms of our detections is that when you plotted the mean change for psi across the experiments, they have very high correlation. So that says that at least the direction, the fold change, is reproducible. So here's just an example of this one specific gene, one specific isoform before knockout and after knockout, you see a difference in their psi distributions. So we have different. So I just want to wrap up. Okay, so the take-home message is that we have this pipeline for correcting mistakes in the UMI scattering, as well as harnessing the UMI to correct for misalignment and other, truncation and other issues in single-cell long-read sequencing. And that allows us to have better isoform quantification at a single cell level. And then with that, we can do some very basic downstream analysis, basic questions, but I think that are not yet answered well. One is to quantify the degree of single cell versus intracell versus intercell splicing heterogeneity through this very simple beta binomial model, as well as to compare across cell populations in terms of their splicing distributions. And with this, we found a universally high intracell splicing heterogeneity, as well as potential targets for splicing factors. I think that's all I have. Sorry, I might go a little fast. I think I think that's my last slide, so I can answer questions.
Okay, thank you so much for the interesting talk. I think if you have any question, please just type that in the Q&A session or just raise your hand. We will try to unmute you. I think there's one question in the Q&A session from Inzo. I'm not sure whether Nancy, you can read that. Or,
I think I have to stop share to read it. Let me
well, I
stop.
can.
Oh,
Well,
I see it. I have this. Yeah. Oh, I don't see it.
Yeah,
Webinar
maybe we
announced.
can read it out for you.
Yeah.
So
Okay.
the question is, in evaluation, it seems that the gene numbers varies a lot. So do you have any explanation for that? So inside she or he is a little worried about the gene sizes that may have the effect on the evaluation accuracy since the existence of truncated RNA.
Oh, gene sizes or gene numbers?
I guess when she or he say gene size, maybe he probably means the numbers of genes.
Okay, yeah. So thanks for that question. One thing that I didn't really mention too much is that we worked with both full transcriptome data sets as well as targeted data sets. Targeted is, I actually think that targeted sequencing is really where we should go to do where we should spend our energy in terms of single cell long read sequencing. Because when it's forced transcriptome, the single cell libraries are so uh, so low in count, right? So then you can't actually profile the isoform expression for many genes very well. So in our data, in both the real data as well as our simulated data is simulated based on the real data. So they sort of mimic those numbers. We have both full transcriptome and targeted. So targeted, there's only going to be a few genes. Full transcriptome, there's more. If that's the question, I'm not sure.
Yeah, I think Nada have kind of a little bit related question. The question is data PacBio or Oxford Nanopore sequencing? I guess it's an Oxford Nanopore sequencing. I mean, your algorithm is aimed at an Oxford Nanopore sequencing, right?
Yes.
But the next question is, how about the sequencing depth? How many reads per sample are great enough to get the decent isoform quantification? That's a good question.
Ooh, that's a hard question. And I'm not sure if I can answer it because it depends on the expression level of the specific gene you care about, right? We've actually underperformed, I think, in most of our full transcriptome sequencing data sets that we had when we were writing this paper, in the sense that the full transcriptome sequencing means you don't target anything, you just sequence a full transcriptome. And there, just with a reasonable budget, you can only capture those really highly expressed. You can only reliably estimate this isoform expression for the high expressed genes, right? For the low expressed genes, you don't get that. Of course, it also depends on cell number. So you can imagine, you have many cells, and then you sort of aggregate across cells. We didn't have a high cell number either. So there's a lot to be desired right now in terms of the state of the data. With targeted sequencing, then we can get enough counts per gene for those targeted genes, and that was much better. We can actually say something meaningful about their splicing and isoform expression. So in terms of what is the minimum coverage required? It depends on what gene you're looking at, right? Whether your gene is highly expressed or lowly expressed, right? If it's lowly expressed, I will say there's no hope unless you do some targeting. And also it depends on what your goal is, whether you're trying to look at like cell splicing, like if you want to look at intra-cell heterogeneity, right? So whether two isoforms are spliced within the same cell. That's much harder. That requires has higher demands on the depth, because you want to have enough, at least two reads, right, for that isoform within the cell. That's, whereas alternatively, if you just want to look for, cell state informative splicing or spatial domain informative splicing, there you could actually pull data across similar cells to increase your coverage, right? So, it's more forgiving. So I think I haven't answered your question, but that's the best I can, tell you about that.
That's great, Nancy. I do have one question I think very interesting to me, but I guess you didn't do it yet in the paper, but I do want to ask. I think one thing very cool in your software. First of all, I think your software, the design is really smart. It's just because you have very deep understanding of the multiple sequences, so that you can design the algorithm so smart to use these three steps. All very smart design. But I think what's cool is your algorithm can directly analyze the intracell heterogeneity versus the intercell heterogeneity. And what's very interesting is different genes show different heterogeneity. Some gene you show is almost always one versus one heterogeneity in all the cells, or some gene or some exon. Well, some other exon or other splicing sites, they tend to be either 0 or 1 for the heterogeneous value, which means there is no intracell heterogeneity, but great intercell heterogeneity. So what I'm wondering is why. So did you try to explore what might be the either biological mechanism or biological reasons, such as maybe different regulator cause different type of heterogeneity? Or do you think it could be, I think it's unlikely, but do you think there might be some technique effects there? Maybe the sequence error are biased towards some gene, some sequence, so that your algorithm is very sensitive at some gene, but less sensitive at some other sequence, other genes.
Yeah, I think, I'm very intrigued about that as well. And I don't think we have answered that question. But I have to say first, the algorithm, a lot of that is due to Yun Qian. I have to ask him before this talk what exactly he did. So, he's designed this algorithm. In terms of why one thing I don't know if if was this true. We found that a lot of the intracell heterogeneity events we found were probably, I don't know, were alternative 3' or 5' UTR, right? Something, there was a lot of intracell heterogeneity in that, in the last UTR usage. So biologically, what does that mean? I don't know. But I think it is important to distinguish between these two types of alternative splicing. One is where you have alternative splicing event that defines new cell populations. The other is where multiple splice variants of the gene are both fundamentally kept in the cell, right? They're made and kept in the cell, so there must be some functional relevance. So Yeah, we don't know. We don't know what is the true biological reason of it yet.
I think
Yeah.
I
I
can.
think you,
Oh, sorry. Go ahead.
yeah, please go ahead.
I think I have some insights about this question. I haven't gone through all of those genes with high intracellular heterogeneity, but for several of our examples, I do check their functions. For example, one of the most representative, which is a SIRBP1. This gene is actually about the microtube, so which encodes for protein to build the skeleton of the cell. And you know that the microtube is actually a very dynamic structure, so cells frequently structured and unstructured, and it get involved with multiple types of proteins to build the microtube. So I think it's very important for cells to maintain the expression of multiple types of isoforms to maintain the dynamics of the cell skeleton. While for those high intercellular heterogeneity genes, for example, the PKM1, it denotes that cells, they chose different isoforms for very different mechanisms. For example, PKM1, the different isoforms of PKM1, they did intraheterogeneity and high interheterogeneity. One of the high intracellular heterogeneity may denote both of them. They are very functionally important to maintain the regular cell dynamics, while high intercellular heterogeneity may denote some kind of tendency for some very specialized mechanism of that kind of cell type. That's my intuition for this, but I haven't gone through all genes for with high intracellular heterogeneity.
That's actually very, very wonderful insights. It does provide those biological, importance there. So yeah, I think you do one of your results did show when you knock out one regulator, the distribution of the heterogeneity or the altered. So I'm just wondering, I'm not sure whether it's possible to do it, but if you have all these, let's say if you have 500 high intercell heterogeneity size and 500 intracell heterogeneity size, then you try to analyze, their enrichment in the targets of each regulator. That might provide some, biological clue to explain it.
Yeah, I think that's a good idea, like to look at the distribution of this phi, which is high phi is high intercell, low phi is high intracell, and then
Right.
look at how that this varies across pathways and across regulators. I think the
Yeah,
data has
maybe
become
just for
much
your
better since
maybe just
we,
for your, maybe just for your future project. I mean, in this paper,
yeah.
I know I say you already have very comprehensive lots of results there.
Yeah, I think the data has become better since we published this paper. So I think there's a lot more opportunities now with the data that's being generated now as compared to when we were doing this paper.
Cool.
Yeah.
Great.
Yeah.
I think that's a great discussion. Do we have any other question from the audience? All right. Well, we run a little bit more time. I think we're a little bit late, but I think it's very good discussion, very good presentation. Thank you so much for Dr. Nancy Zhang and also Kaifu for co-hosting this seminar. And I hope you two have a great day. And thanks everyone for attending this seminar. Thank you.
Yeah, thank you. Thank
Thank
you,
you,
Yang
thank you,
and Kaifu
Nancy.
and everyone. It's good to see you. It's
Yeah.
Bye-bye.
Bye. Okay, can we stop recording?